Good evening. We will be beginning in a couple of minutes. Let's wait for everybody to join in and then we can start. So till then, I'll just be switch my camera off for a few minutes. I'll try to upload the slides. Okay, I think we can start. So for that, I'm just preparing my teams. Just give me a moment. Okay. My screen is yet not visible. Um, Let me try once again. Now it's visible. So, welcome to the fifth session of this course, which is on the purification techniques which are used for enzyme production. So, this is the third part of enzyme production that we will be discussing about. So, we have already talked about how plasmids are used, how they are overexpressed, and different tags which are used with different expression systems that are used. We have talked about it. Now we will be talking about purification techniques. So, briefly talking about the upstream, the enzyme production entire schematics, there is an upstream process which involves cloning, transformation, and overexpression. Then there is a downstream process that involves, either this as an extracellular product or an intracellular product. So intracellular product, if formed, needs to be disrupted. So we need to perform cell disruption. There are various ways of doing this, either by chemical method or enzymatic, mechanical, or physical, with which we get a crude product. For extracellular product, we'll directly get a crude product. Then there is solid liquid extraction, which one can be done by centrifugation, extraction, or filtration. Then we go for concentration. So for that, we can go for evaporation, ultrafiltration, or precipitation. Then we go for purification using chromatographic techniques. So that is the main thing. And then we get the final product, fine, which is the enzyme. Now, talking about the cell disruption methods, there could be physical methods that plays with physical parameters to damage the cell, right? So there could be, for example, thermolysis, which can be used only for thermostable products. So here, heat is used to kill the organism and disrupt its cellular integrity. Similarly, the effect of heat shock depends on the ionic strength, presence of chelating agents, like, for example, EDTA, and the presence of proteolytic enzymes. Then another way of doing is this is osmotic shock. So in case of osmotic shock, what happens? We give a prolonged exposure of cells to a hypotonic solution. Fine. It causes an osmotic imbalance, causing cell lysis. There's Hof's equation. So pi is equal to RT Ci minus Co. So Ci minus Co is the concentration of solute inside cells minus outside cells. So that is the main driving force behind an osmotic pressure, pi. R and T, R is the universal gas constant. And T is the absolute temperature, that is in Kelvin. So each cell has a different, kind of susceptibility towards osmotic shock. So for examples, RBCs, they can be easily lysed. But if we talk about plant cell or animals or bacterial cells, they have a very thick cell wall, and osmotic imbalance usually is not sufficient to break the cells. Another very preferred method is sonication. In sonication, there is ultrasound waves which are greater than 20 kilohertz. It causes cell disruption by cavitation. So it's how it works, and the interaction of ultrasound with liquid causes frequent compression and decompression. These shock waves cause cell lysis. They can also generate heat, hence the sample needs to be in ice usually when we perform sonication. There are some chemical methods also, enzymatic methods. Where we can go for alkali treatment, which causes lipid saponification, and it disintegrates the membrane, cell membrane essentially. We can also use detergents, which causes solubilization of lipids, and they form micelles. Now, the concentration at which these micelles are formed, that concentration, the minimum concentration is called CMC, or critical micelle concentration. For a particular detergent. Examples are like SDS, then we have Triton X, saponin, digitonin. These are all detergents. Now, talking about permeabilization, this is achieved by organic solvents like toluene. Use of organic solvents would permeabilize the cell membrane. That's the idea. So it causes swelling of cell wall, swelling of cells, finally causing their rupture. And this is all done if we want to get this protein in from the intracellular fraction. If this protein is secreted, then these problems don't are not there. So then we can obviously go for enzymatic digestion by using enzymes. So lysozyme is used, very commercially available. Other bacteriolytic enzymes are glycosidase, acetyl muramyl, L-alanine uh, amidase. There are certain proteases which can also affect the bacterial cell wall. Similarly, for yeast, we have glucanase, protease, manganese or whatever, and chitinase. For plant cells, we have cellulase and pectinase. Right? So these are all the enzymes that are used for cell lysis. The mechanical methods which can be used is either by bead mill disruption. So there's an instrument called bead beater where the beads are made up of either silica or glass, or it could be zirconium oxide. These are used to make the beads, and these beads are used then to lyse the cells. Then there is high pressure homogenizer. High pressure homogenizer, it uses a high pressure displacement plumb. And there's a small cavity from where your cells go out and they get lysed. So this is the principle of an instrument called French press for cell lysis again. Then after this, we go for chromatography. And very simple, I mean, the basic principle of chromatography is to separate individual components from mixture. And we would always look at the efficiency of a chromatography through something called as the distribution coefficient. Uh, Also marked as Kd. So it is if in two phases, concentration of molecule in phase A, or maybe the matrix, and the concentration of molecule in phase B, that is the buffer. It could be for a molecule, column, or bead, whatever you are using. So in column chromatography, there is a stationary phase, which is the column. And the analyte, so if this is the cylindrical column, the analyte is loaded from the top, it passes through the column, and you get uh, uh, the eluent. So this is the eluent, this is from where you give the analyte or the sample, whatever you call it. Okay. So if you plot the absorbance versus the elution volume, that kind of plot is known as a chromatogram. Elution volume. So it will look something like this. Okay. So here you would inject sample, and based on your chromatographic techniques, you'll get different peaks, right? The resolution of a chromatographic technique or a chromatographic column is based on how two different peaks are separated. So peak can be like this. These are known as fused peaks, or they could be distinct peaks. That will give you the value of resolution, which is calculated as the difference in retention time by the average width uh, of the baseline, right? So if the RS value is like 1, then the peaks are separated by like 97.7%. If the RS values are greater than 1.5, then we call those kind of column as a good column, right? It can be used. So in a chromatographic system, what are the components? So first we have the solvent reservoir, then a pump, mixer, then we have a sample injection loop, which will inject the sample. Then we have the entire column. Then we have a detector. which detects the UV absorbance or whatever is there, what we are getting from there. And then we have a fraction collector, which collects these fractions. And then we have the recorder, which is usually a PC kind of situation, which will basically plot the entire thing. Now, what are the different forms of chromatography? So there could be partition chromatography. So where the analyte is in two phases. One is the liquid and stationary phase. Another one is the mobile phase. Fine. Similarly, we have adsorption chromatography. What happens in adsorption chromatography? Essentially, a matrix can hold analyte on surface through mutual interaction due to different types of forces. These forces could be hydrogen bond, they could be electrostatic interaction, they could be van der Waals force, something like that. So examples of adsorption chromatography are like ion exchange chromatography. Then there is hydrophobic interaction chromatography, affinity chromatography. So they are all uh, based on the principle of some sort of interaction between the analyte and the matrix. Okay, that's the point. Now, if we talk about different proteins in general, they have several properties which can be used for different chromatographic techniques. So they have affinity towards a particular metal or an antibody, for example, that can be used for affinity chromatography. Proteins also have hydrophobicity, that is, they have hydrophobic patches. They can be used for hydrophobic interaction chromatography. They have a surface area and they form a 3D structure which has its own molecular mass and everything. They can be used for gel filtration chromatography. We'll talk about it. And then there is charge also. Charge is usually present in a protein that can be used for ion exchange chromatography. So this is the essential, the parameters or the properties that proteins have. Now talking about ion exchange chromatography, if we talk about it, then ion exchange chromatography distributes the analyte as per charge. So let's say if your matrix is positively charged and your protein is negatively charged, they will bind. That's the idea, right? Um, so there are like the ionic strength of, it is dependent on the ionic strength, the pH of the buffer. So ion exchange could be also further divided into two types, either cation exchange or anion exchange. So it is based on the analyte. So if the cation is there, cation is the analyte, then we call it as cation exchange chromatography. If the analyte is anion or negatively charged, then we call it as anion exchange chromatography. So based on this, there are several, you know, columns which you can read about. So based on that, so these columns are basically the matrix which is there based on different, whether you have a strong analyte, strong cationic analyte, or strong anionic analyte, or something like, or weak. In based on that, we have different column matrices. So how to select matrix? So for that, you need to know the pi value. The pi is isoelectric point of a protein that you need to know, and the net charge. Fine. Then you need to know the structural stability of the protein and the enzymatic activity. So how to calculate pi? There are several softwares to do this. For example, in ecstasy, we have a server which can calculate the pi of a protein through individual amino acids, through its sequence, basically. So if you look at the operation of an ion exchange chromatography, so the column material should be inert. Should not be a reactive material. The mobile phase, it's very important to know the pH and the ionic strength of the mobile phase, right? These are very important. For sample preparation, samples are usually in the mobile phase. In the mobile phase, we have the sample usually. And elution can be done in two ways, either by a stepwise gradient or a continuous gradient. So then what we do after this is done, we go for column regeneration, where the column is regenerated for the next cycle of chromatography. Then there is hydrophobic interaction chromatography. It exploits the property that proteins have hydrophobic patches which can only interact with hydrophobic moieties. So that's how it does. So if we look at the solubility versus salt concentration curve, we would see something, a peak like this. So at higher salt concentration, we would see something called salting out. And at lower peak concentration, we will see salting in. At an optimum pH, the solubility of the protein will be highest. So that's the idea. So there are then separate, you can study about the slides where it talks about different matrices that can be used for different functional groups. If we talk about the operation, first we need to equilibrate the column. Equilibrate the column, and that is usually done with ammonium sulfate. Then we go for sample preparation. Again, it is injected usually. Then we go for elution. So elution can happen by decreasing salt concentration or changing the polarity. Or we can also use detergents. Detergents would actually change the hydrophobicity. Even the hydrophobic moieties might expose themselves or something like that, in detergent, because micelles can be formed. And finally, what we do is column regeneration. In this case, we use 6 molar urea or guanidine hydrochloride, something like that. And column can be stored in 4 degrees in the presence of 20% ethanol and 0.05% sodium azide. Then comes gel filtration chromatography. In gel filtration chromatography, the principle is the sieving effect. So if there are, let's say, large pores in the sieve, there could be large pores and there could be small pores. Okay, so this is how the sieves work. So there is exclusion of large molecules and inclusion of small molecules. So based on this, you would see when you will plot the chromatogram, if there is absorbance versus the elution time. If you see three peaks, the largest molecule will be here in the first peak. Then you will have a medium-sized molecule, and then you will have the smallest molecule coming out at the end. So in a column, in the matrix, it has the sieving effect inside, and the analyte is distributed based on its molecular size. Also, this is the reason why gel filtration chromatography is also known as size exclusion chromatography, simply based on its principle. So these terms can be used interchangeably. Okay. Again, you can look at the different choice of matrices in different, that is based on different the molecular weights that you are trying to look at. And then there are certain other problems like, troubleshootings could also be there, like the back pressure problem, which can happen due to the improper packing of the column. There could be clogging because a column is run for a long period of time. It can clog the pores. Yes, that can happen. There could be precipitation of proteins that can happen. So these are the problems that can happen while doing gel filtration. And there should not be any particles as insoluble particles usually should not be present in the sample. Otherwise, they will very easily clog the column. So that's a problem. Apart from that, the fourth kind of chromatography is affinity chromatography. So in this case, the most commonly used is the nickel NTA, nickel NTA chromatography. So it has the beads which has nickel or even cobalt in certain cases. That has high affinity towards histidine. It's an amino acid. So usually you must have seen in pet vectors, there are polyhis tag in a protein. You tag the protein with histidine, and this will basically bind to this the nickel. And that can be used, this affinity function can be used to purify your protein of interest in this way. Other affinity chromatographies can also be performed using antibodies against your protein. It will be very much more specific, but it's again very costly as well. So with that, I think we are done with the discussion. Now we will look at certain questions basically. So the organic solvent used to disrupt the plasma membrane is toluene, acetone, acetic acid, or hexane. Any answers? Anybody would like to answer? Sorry, I can't hear you. Hello. Okay, toluene will be the answer. Osmotic pressure generated in solution is by the difference in solute concentration, salting out, salting in, and same solute concentration. So we already know that the pressure, osmotic pressure can be written as, I mean, the formula for this would be RT into Ci minus Co. That is the difference in solute concentration inside and outside the cell. Osmotic shock is used to remove the nucleus, cytosol, cytoplasm, or periplasmic substance. Okay, so the answer to this would be periplasmic substance. The chromatographic technique that depends on highly specific interaction between pairs of biological materials such as enzyme substrate is affinity chromatography, ion exchange chromatography, gel permeation chromatography, or adsorption chromatography.
Absorption chromatography.
If we are talking about very specific interactions, so let's say enzyme substrate, or let's say antibody and its corresponding antigen, something like that, it comes under affinity chromatography. Actually, you're right, adsorption chromatography is the broad umbrella term, uh, which includes affinity chromatography as well, ion exchange chromatography as well. But when we are specifically talking about specific interactions, which it means affinity chromatography. So it is a little bit confusing question, although your answer is also correct. Dash allows chaperones to recognize a wide variety of misfolded structures. ATP hydrolysis, GTP hydrolysis, ATP binding, or ATP release. Chaperones are basically, these are molecules which assist in protein folding. So they recognize, misfolded proteins, and they go there and they fold the proteins. And for that, they require ATP. So ATP hydrolysis is the process. The interaction that contributes most to protein folding is covalent interaction, hydrophobic interaction, ionic interaction, or van der Waals interaction. Any answers?
Hydrophobic interaction.
You're absolutely right. Absolutely right. Chimerized humanized monoclonal antibodies are generated using hybridoma technology, recombinant DNA technology, southern blotting, or immunization.
Hybridoma technology, maybe.
You're absolutely right. Question 8, match the following. The receptor is avidin with what would, okay, so poly A, lectin or heparin, and the affinity towards protein ligand would be DNA binding site, glycoprotein, biotin containing enzyme, or poly U mRNA. So avidin has affinity towards DNA binding site. Fine. Poly A will definitely wait.
Poly A, it is the binding sites, maybe, I think. DNA binding sites.
No, actually not. I'm trying to figure out. Um, if the receptor is poly A, it should bind with poly U, right?
Poly U mRNA.
Isn't it?
Oh,
So because A's and T's are complementary to each other, so A and U are also complementary to each other. So I think that would be the and avidin, streptavidin, all these things are for biotin containing enzymes. I'm sorry, it's not for DNA sites. Uh, Lectin is for glycoproteins, and heparin is for DNA binding site. I think this will be the answer. A3, B4, C2, D1. Question 9. Dash is not a highly specific biological interaction to be used in affinity chromatography. Is it enzyme substrate, antigen antibody, receptor ligand, or cation anion, which is not a highly specific biological reaction? Any answers?
Enzyme substrate.
Sorry?
Enzyme substrate.
Enzyme
It's not.
substrate is specific. It's very specific. Antigen antibody is as specific as well. Receptor ligand is also specific. What is not specific is cation and anion. Any cation, any anion can bind together. It's basically the principle of ion exchange, if you remember.
Yes, yes.
Yes. Dash tag is a 6 amino acid residue peptide which can be immobilized by transition metal ions. Is it S tag, his tag, MIG tag, or flag peptide?
Which tag?
Yes, you're right. Histidine tag, and then the transition metal ions are like cobalt or nickel. Two monomeric proteins are migrated identically in SDS page but got separated in the size exclusion chromatography. The reason would be they have different isoelectric points. They have the same isoelectric point. One protein has more disulfide bonds, one protein is a monomer, and the other is a tetramer. What would be the answer? I think we discussed something similar to this in our first lecture, if you remember. There also we talked about size exclusion chromatography. We talked how we calculate oligomeric status of a protein using gel filtration. You remember? Okay, so the one protein is a monomer, another one is a tetramer. So when we do an SDS page, we denature the protein, right? So every protein will be in its monomeric form. It will not have any other interaction. But when we are doing gel filtration or size exclusion, we will get the oligomeric status of the protein, right? That's the idea. A globular protein of molecular weight 50 kDa or kilodaltons exists as a mixture of monomers and dimers in a solution. The most appropriate technique for separation of these two forms of protein is thin layer chromatography, paper chromatography, ion exchange, or gel filtration. Just discussed about it. Answers?
Gel filtration chromatography.
Yes, you're absolutely right. The procedure in which cell lysis occur because of shock waves which are formed due to collapses occurring in buffered solution containing cells is referred to as alkali treatment, osmotic shock, ultrasonication, or detergent treatment. Ultrasonication, if you remember. Yeah, you're right. A digestive enzyme, salivary amylase in saliva, begins the digestion of carbohydrates, proteins, fats, or nucleic acid. Amylases usually degrade amylose and amylopectin, all these kind of things. So these are usually sugars, they are carbohydrates.
Carbohydrates.
In electrophoresis, if the buffer pH is above the isoelectric point of the protein will migrate towards cathode, migrate towards anode, not migrate at all, migrate partly to anode and partly to cathode.
Migrate partly to anode and partly to cathode.
No, actually not. So isoelectric point is the point at which the charge of the protein is neutral, right? At a higher pH, a pH higher than the isoelectric point, uh, the protein has a positive charge, right? If it is less than, sorry, it has a negative charge, I think. Sorry, not a positive charge, it has a negative charge. pH less than pi, then we have a positive charge. So a negative charge will move towards the anode. Clear?
Okay.
Okay. So with this, we are done with today's session as well. I think if there are no further questions, we can. Oh shit, I just discarded. Okay. I might have to write that again. But anyway, that's fine. Should have saved it, but If there are no further questions, we can call it a session and we can maybe meet again next week, the same time. Are there any further questions?
Actually, I have on those things.
Okay, ask.